Hi, this video is a video that goes through some exam questions on homeostasis, specifically the homeostasis and control mechanisms for glucose concentration in the blood. Okay, so this first question reads like this. Broken bones are repaired by cells called osteoclasts and osteoblasts. Osteoblasts secrete a hormone called osteocalcin in an inactive form. Osteocalcin is a protein. The active form of osteocalcin binds to a receptor on the beta cells in the pancreas, stimulating the release of insulin. Osteoblasts have receptors for insulin. The diagram shows how the production of osteocalcin by osteoblasts is controlled by positive feedback. Okay, by positive feedback. So that's the first sort of link there. Some of the things to what we've been chatting about. We've got positive feedback. That's important. And the fact that we've got beta cells in the pancreas stimulating the release of insulin. Okay, so the question says, the secretion of osteocalcin in an inactive form by osteoblasts is controlled by positive feedback. Okay, by positive feedback. So the secretion of that, use the information from the diagram to explain why this is positive feedback. Now I want to remind us that positive feedback is about how producing a hormone, then that excretion of that means that it exacerbates and makes it release more of the same, rather than negative feedback. So the release returns it back to normal. The further away from normal produces more, which continues to further away from the normal. Okay, so positive feedback. Let's start off by saying, and get some marks here on the answer. So we've got osteocalcin. Let's make sure we've got that as small as we can. So osteocalcin. Okay, so osteocalcin causes, that's important, more, I'm going to highlight that as an underline it, more insulin to be secreted. Okay. Now, on top of that, so that's where you've got a 1 mark in there. And the next mark, we would say that more insulin now if we watch the diagram, so the osteocalcin is secreted, okay, it stimulates the beta cells, okay, to release the insulin. So we're saying osteocalcin causes more insulin to be secreted. However, more insulin stimulates osteoblasts to release more inactive osteocalcin. So more insulin stimulates more secretion of osteocalcin. So that goes through that. The idea of more is required. So because we want more, we need more of this stuff. Okay, it exacerbates that whole process. So more and more. So osteocalcin is then formed more insulin, which forms more osteocalcin, which forms more insulin. Okay. So the next question says the acidic pH conditions created by osteoclasts cause inactive form of protein osteocalcin to change into active form. And it was the active form that was needed for the stimulation of the beta cells to make insulin. So let's suggest how. Now this is a question of our understanding of proteins. So we go back into our understanding of proteins and say that changes in pH causes changes to the tertiary structure of the protein. Okay, so changes in the pH causes changes to the tertiary structure of the protein. Okay, and why it does that is because the pH changes causes. So why did it do that? Because the pH changes causes the polar hydrogen bonds to break, which goes back to, so it really, that's probably the first mark. The pH causes the polar hydrogen bonds to break, which in turn changes the pH, which in turn causes a change in the tertiary structure of the protein, because the tertiary structure is held in place by those hydrogen bonds. Okay, so those are the two marks. First mark there, second mark there. Next question. Binding of insulin leads to an increase in the rate of respiration in cells, such as osteoblasts. Okay, explain how. Okay, and it's worth 2 marks. Now this takes us back to our understanding of what happens when we release insulin. So insulin, okay, reminding ourselves, so insulin and if you can remember, it's called tyrosine kinase. And what that does, so insulin binds to tyrosine kinase. That causes the molecules, the vesicles, causes, if you can remember, we drew this, these structures that have these protein channels within them. So we've got we've got a this green is going to represent my bilayer. Okay, we've got a vesicle, okay, like this, which has this vesicle has these protein channels for glucose. So insulin binds to the tyrosine kinase, and when the insulin binds to the tyrosine kinase, okay, it causes the production of extra glucose protein channels Okay, so it causes production of extra glucose protein channels. Okay, more of them. Okay, So if we've got more protein channels, more glucose can now enter cells to undergo glycolysis. You could say, I think if we just said for respiration, that would also be okay. So we're saying the insulin binds the tyrosine kinase, which causes the changes, which means the vesicles then fuse with the membranes. Those fusing membranes leave these protein channels for glucose in there. That causes extra glucose to enter the cells. More glucose can now enter the cells and undergo glycolysis for respiration. And that takes us to the end of question